Meiosis is a type of cell division in sexually reproducing organisms that results in 4 daughter cells, each with haploid sets of chromosomes. Meiosis follows 2 of Mendel's laws, the law of independent assortment and the law of segregation. The law of independent assortment states that 2 or more cells sort independently, meaning that they segregate individually during gamete formation. The law of segregation states that the 2 alleles for a characteristic segregate during gamete formation and end up in different gametes. During interphase, the chromosomes are uncondensed in the nuclear envelope, and the chromosomes replicate into a pair of duplicated homologous chromosomes. This results in 2 centromeres, a region in the DNA responsible for attachment of the spindle fiber. Prophase 1 is the longest stage of meiosis. The chromosomes condense progressively throughout prophase 1. The nuclear envelope fragments as well. Each homologous chromosome loosely pairs up and is aligned, gene by gene, as crossing over, or the exchange of genes between homologous chromosomes occurs. Each homologous chromosome has 1 or more X-shaped regions called chiasmatas, where crossovers have occurred. Later, in prophase 1, spindle microtubules from one pole will attach to the 2 kinetochores of a homolog as the homologous pairs move toward the metaphase plate. In metaphase 1, pairs of homologous chromosomes line up at the metaphase plate, where they face one another and form a tetrad, a group of 4 chromatids. Both chromatids of one homolog are attached to kinetochore microtubules of one pole. Those of the other homolog are attached to microtubules of the other pole. During anaphase 1, proteins that are responsible for sister chromatid cohesion along chromosome arms are broken down, allowing the homologs to separate. The homologs move to opposite poles with the help of the spindle apparatus. Sister chromatid cohesion continues at the centromere, causing the chromatids to move as a unit toward the same pole. At the start of telophase 1, each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome still consists of 2 sister chromatids. Cytokinesis, the division of the cytoplasm, occurs simultaneously with telophase 1, forming 2 haploid daughter cells. In animal cells, a cleavage furrow forms, and in plant cells, a cell plate forms. Between meiosis 1 and meiosis 2, there is no chromosome duplication. In prophase 2, a spindle apparatus forms. Toward the end of prophase 2, chromosomes move toward the metaphase 2 plate. In metaphase 2, the chromosomes line up at the metaphase plate. Because of the crossing over that had occurred in meiosis 1, the 2 sister chromatids of each chromosome are not genetically identical. The kinetochores of sister chromatids are attached to microtubules extending from the opposite poles. In anaphase 2, the proteins holding the sister chromatids together at the centromere break down, allowing the chromatids to separate. The sister chromatids move toward opposite poles as individual chromosomes. In telophase 2, nuclei form. The chromosomes begin decondensing, and eventually, cytokinesis occurs. One parent cell produces 4 daughter cells with a haploid, unreplicated set of chromosomes. The 4 daughter cells are genetically distinct from one another and from the parent cell. The end product of meiosis is either a single egg cell, or in this case, 4 sperm cells. This is because in female gametes, 3 of the 4 egg cells experience apoptosis. Meiosis is now complete.